Hello, students. For Tuesday lesson, we are going to learn. We are still actually under topic number 15. Okay, still actually topic number 15 under control and coordination. But this time around, we're going to focus on control and coordination in plants. Okay, so we're going to look at describe the rapid response of Venus flytrap, which is a type of carnivorous plant, to stimulation of hairs on the lobes of modified leaves, and explain how the closure of the trap is achieved. So the syllabus focuses on closure, okay, so not open, okay, only on the closure of the trap, and it uses the term explain, yeah, and that is very very important. So normally any keyword that involves the word explain, it involves a lot of detailed mechanism. Okay, I have given you the link, yeah, for today's lesson on the Venus flytrap, just to give you a prior knowledge, okay, on what uh, Venus flytrap really is. So basically, a lot of us think that electrical stimulation of action potential only occurs in animals, but actually it also occurs in plants. So this action potential occurs in plants, which was discovered by Charles Darwin, yeah, long time ago. Um, and he observed, by observing the Venus fly trap. Right, let's just look at some of the backgrounds on electrical stimulation of the plant. Uh, so basically, they also have resting potential, they also have action potential, but they don't have neurons like in animals. Yeah, so plants do not have neurons or nerve cells. So rather, they travel along, and this is a very important point. Okay, this is a very very important point. So if they don't have neurons or nerve cells, how do the action potential travel? So the action potential travel along cell membrane, along the cell membrane of cell membrane. Okay, they travel along cell membrane of plant cell and from cell to cell through plasmodesmata that are lined by cell membrane. Still plasmodesmata, but aligned by cell membrane. So in plants, action potential travels along cell membrane or cell surface membrane. And then there's another difference. The action potential generally much longer, so it's and travel more slowly. Yeah, for example, in perhaps in animals, it's in millisecond. Yeah, in animals it's in millisecond, whereas in plants like the Venus flytrap, I think it's within second, as you can see here, second and second. Okay. So in animals, it's within milliseconds, whereas in plants, it's within second. Okay, not millisecond. Okay, all right. Right. Let's just go straight to. There are actually lots of different types of plants that, that can trigger action potential. But the ones that we are only interested in, which is stated in outline in the syllabus, is the Venus flytrap. Okay. So this is how it looks like, the Venus flytrap. I hope that you have watched the video. Okay, the Venus flytrap actually looks small. It's a very small plant. Okay, about medium size. Okay, it's not really that big. It's not really that small, but it's a medium size. Um, it's a carnivore. Okay, it feeds on insects mostly, uh, and they're actually found somewhere in USA. Yeah, in America, in the North Carolina and South Carolina. Okay, and particularly in this region here. Okay, used to be here in this region. Okay, so what do we know about this Venus flytrap? It's a carnivorous plant. Okay, it obtains supply of nitrogen by consuming insects. Okay, because insects have proteins, and so they can get the nitrogen from digesting insects. It has two lobes, okay, two lobes, and in the middle it has a midrib, or also known as the hinge cells. Inside of each lobe is often red and has nectar around the edge. So the nectar is around the edge. It's around in this region here, okay? The nectar is around this region here, around the edge, around this region here, okay? Each lobe has three stiff sensory hairs that respond to being deflected, okay? So here we have 1, 2, 3, yeah, and then 1, 2, 3. These are all sensory hairs. Outer edges of the lobe have stiff hairs that interlock to trap insects. So this is the like interlock. It's actually called cilia, okay, but it's a strong one. The deflection of says here, the deflection of sensory hairs on the inside of the folded leaves stimulate action potential that travel very fast across the leaf, causing it to fold over and trap the insect. Okay, so just some features of the leaf. So this is the inner epidermis, this is the outer epidermis, these are the sensory hairs. On each lobe there are 3 sensory hairs, and then these are the midrib, yeah, or hinge, which is under tension. Okay, normally a trap, when it is open like this, okay, this is under tension, just like when you set up mouse rat trap, yeah, or mouse trap. Okay, it's always some part of the device is always under tension. Okay, and for this particular trap, this area here is the one which is under tension, the midrib. Okay, the midrib. Okay, so just so the nectar is around here. This is the around the edge, yeah. This is the nectar. Okay, and then use a black color now. This is where the digestive enzymes are released. Yeah, it's this in this area here. This is where the digestive enzymes are released. Okay, digestive enzymes, meaning when the insect is trapped in this region here, the enzymes will be released, okay, by exocytosis. Okay, of course it's using vesicles. Will the vesicle will fuse with the cell membrane and then will undergo exocytosis, and it will release the enzymes, okay, to the outside to digest any trapped insect. Okay, so hinge or hinge, I think we call it hinge cells, is also known as the midrib cells. Okay, so now we're going to go look at this part here. So when it's open, it has this convex shape, yeah, convex, like this, convex. Okay, this is a convex shape, convex. Okay, convex shape, and the opposite is like this. So this is concave. Okay, so just to recap, all these different shapes. So at the moment it's convex. Okay, convex, so it's open, so convex. The lobes of the leaf bulge upwards, so bulging upwards when the trap is open. And remember, this hinge region is under tension. Remember, any trap, when you set up a trap, there is always some part of the device that is under tension. Okay. So this, this like they call it the stiff hair. Outer edge of the lobe have stiff hairs that interlock. So stiff hairs here is also called cilia. Okay, so cilia or stiff hairs. This hair or the cilia is different from the sensory hair. The sensory hair is different from the cilia or from the stiff hairs. Okay, so here's how it looks like. And this is as well another one here. Okay, so from here you can see the nectar. Okay, this is the nectar around this region here, nectar. Okay, and then this is the digestive enzymes. So this is the digestive enzyme here where the trapped insect will be digested. Okay, so who knows, a question may come out where you have to label where the, but according to the syllabus, it doesn't say that you have to label. Yeah, it says here, describe the rapid response of the Venus flytrap to stimulation of hairs on the lobes of modified leaf, and explain how the closure of the trap is achieved. So it's a bit demanding actually. Yeah. Okay. So when the leaf is closed, the lobe bulge downwards. Okay. So bulge downwards mean, this is upward, yeah. And if it bulge downwards, then it will create the closure like this. Then it will close. Okay, it will close. So initially it's a convex. Of course, it's on the other side, it's convex. And then it will be concave, yeah, when it is closed. Okay, so now this is very, very important in the next few slides. So when this hair is deflected, okay, of course it will take the first hair and then the second hair. Yeah, if one hair is deflected, let me just explain this again. If you watch the video, that's very good. So if, let's say, this hair is deflected, okay, then if the second, this is the first one, hair is deflected, and if, so the time starts, start time, okay, from 0 second, 1 second, and so on. If the second hair is deflected within about 30, so I'm just trying to, because it depends on the textbook. Sometimes between 20 to 30 seconds, yeah, meaning the first hair deflected, the second hair deflected within 20 to 30 seconds, then action potential will occur. Okay, so the way the action potential occurs is that it will move, it will cover all this lobe, okay, and then it will go down to this hinge. Okay, go down to this hinge. The same thing here. It will, its potential will travel across the lobe, and then it will reach the hinge. Okay. Okay, that's one scenario. The second scenario is if, for example, this is deflected, the first hair is deflected, and then start time. Okay, and then let's say if it's over 30 seconds, so there is no second deflection. If there is no second deflection after 30 seconds, okay, then it will not close. Okay, it will not close. So let's say this is the first air which gets deflected, and let's say the second hair gets deflected at 40 second. Okay, at 40 second, then this is the time is reset. Time is reset, meaning this is now 0. Time is 0. Okay, this because it is deflected at 40 second, is not within 30 second. That means this is already. That means this is the time is reset, meaning this now becomes the 0 second. So if there is another hair is deflected within the 30 second, then it will close. Okay, so anything beyond 30 second, if anything beyond 30 second, if the second hair is deflected, then the time is reset back to 0. If you understand what I mean, yeah, time is reset back to 0, and then until it waits until it has another deflection within the 30 second. Okay, so basically the action potential, the way the action potential moves is that it moves across this lobe, and then it will reach the hinge. It moves across this lobe, and then it will reach the hinge. Okay, so the hinge is a very critical region of this Venus flytrap. Okay, so here we have this is the first deflection. Okay, the first deflection. So you can see when it's deflected, so this is what we call mechanical stimulus. Because it is deflected and there is stretching occurs, so it's mechanical stimulus, and that causes the entry of calcium. Okay, so that means the cell membrane, all of the cell membrane here, the calcium channel will open, and that will allow the calcium to enter, and that is when the action potential occurs. And action potential occurs, remember, only occurs along cell membrane. Okay. So action potential occurs along cell membrane or cell surface membrane. Yeah. So so this is the calcium. Okay, this is just to show you. This is actually a screenshot from a video. Okay, so this white color here is the calcium. So the entry of calcium causes action potential which travel along the cell membrane on each lobe. Okay, so action potential here is electrical energy. Okay, mechanical energy converted to electrical energy. Okay, so after the mechanical stimulus, because this hair is deflected, there's a stretching there, mechanical, and then it will be converted to electrical energy. The electrical energy is because of the action potential occurs along the cell membrane here due to the entry of calcium. So if you look back in transport in animals, so transport in animals, for action potential to occur, it is sodium that enters. So this is a very big difference. Okay, so in animals, action potential is due to the entry of sodium, but then in plants, action potential is due to the entry of calcium. Okay, entry of calcium. Okay, now this is actually from a video which I screenshot, yeah, just to make trying to help you understand more about the calcium movement of calcium. And this is the influx of calcium shown in green in color, spreads across the lobe, yeah, when the first stimulus is detected, meaning the first hair is deflected. So when the first hair is deflected, which occurs around this at this time here, okay, first stimulus it says here, this is the first stimulus. So you can see calcium in flux. That means all of the lobe here, the green color shows the concentration of calcium, which is going into the cells of each lobe. Okay. And then after, over some time, the concentration of calcium will decrease, yeah, if there is no second stimulus. So over time, you can see here, just looking at the time frame, yeah. So here we have 0.82 because the first stimulus is is detected, meaning the first hair is deflected about 0.82 or before that. So calcium will enter. Okay, you can see green color there. But if the second stimulus is not detected, you see, then you can see the concentration of calcium decreases. So the green color becomes paler, and it becomes even paler. You see, it becomes even paler until the second stimulus is detected. So now the second stimulus is detected. Okay, so you can see now it's going back again green, brighter green, and this is normally when the second stimulus is detected, the influx of calcium spread across the lobe in higher concentration than in the first one. So in the second stimulus, more calcium will enter than in the first one, and that causes the action potential. Okay, so this is when it's open, and this is when it is closed. Okay, this is from a video which I screenshot. Okay, now this is the most important part here, the mechanism. Okay, mechanism of the Venus flytrap. So we begin with, of course, the nectar will attract the insect, and then there is a deflection of sensory hair. So this is mechanical stimulus. Yeah, this is mechanical energy, which will later be converted to electrical energy. So calcium channels on the cell membrane open, influx of calcium. Okay, let's just give it a tick. Depolarization occurs where cell surface membrane. Yeah, remember in plants, depolarization occurs on the cell membrane or cell surface membrane because plants do not have neurons. Okay, plants do not have nerve cells. So rather, the depolarization occurs on the cell surface membrane or cell membrane. Okay. So the first, when the first hair, this is the first hair, yeah, first sensory hair is reflected. The first one, yeah. There is no action potential yet, yeah, but there is depolarization. Depolarization means there is some movement of ions, like what I mean by depolarization here, the influx of calcium. Okay, the influx of calcium, like this one here, yeah, influx of calcium, but there is no action potential yet. Okay. So this will create receptor potential. This is stated in the textbook as well. Receptor potential is it means that there is movement of ions due to the deflected sensory hair. Okay, but it will not cause action potential yet. Okay, it will only, so when the second deflection occurs, says here, second deflection occurs within 20 or 30 seconds, then depolarization, then that will reach threshold value. Yeah, so the second deflection will reach the threshold, the required strength of stimulus, okay, then only then action potential can occur. So action potential will only occur after the second hair is deflected. Okay, when the first hair is deflected, it's just the depolarization, which means that there's no action potential. It just means that calcium influx of calcium. Okay, influx of calcium, but still not enough to cause action potential. Meaning, still not enough to transmit the action potential to the hinge cells. Okay. Okay, and then here, action potential. So if the second hair is deflected, action potential spread across the trap. It means depolarization spread over leaf, lobe, or lobe to hinge cells. Okay, so it means the action potential will cover the surface of the leaf, and then it will end at the hinge cell, at the midrib or the midrib. And at this point here, that's when the convex will change to concave. Okay, so convex like that, concave like that. Now, for Cambridge, it's important that how does it what makes the convex change to concave? What is the mechanism? Okay, okay, we know that the convex changes to concave because of action potential occurs, influx of calcium, action potential occurs, and so the shape changes. But rather, we want the mechanism, how the convex is changed to concave. Yeah, what is the mechanism? What is the sequence of events? Okay, but before that, let's have a look at this one here. Mechanical energy is converted to electrical energy. So deflection. Okay, deflection is deflection of sensory hair. This is the mechanical energy. Okay, and then the action potential spread. That is the electrical energy. Okay, the 8P, the action potential is the spreading of the action potential across over the lobe, and then later on to the midrib or hinge cells, is what we call electrical energy. So now this is the mechanism how the convex change to concave. Okay, and this is stated in the learning objectives and explain how the closure of the trap is achieved. So that means we have to talk about this one here, this slide here. Okay, so you can see here, when action potential arriving at the hinge cells or midrib, this causes proton to pump out of cell into cell wall. Okay, so if I can, before that, let's just read first. So the pH will drop. That means it's going to be very, very acidic. Okay, and this will break cross-link in cell wall. So the breaking of the cross-link within cell wall, meaning the tension is loose. Okay, loose. It's no longer under tension. So the midrib is no longer under tension because the cross-link is now weakened. Remember, when it's open like this hinge here, okay, this hinge here is under tension. But if it closes, then this is no longer under tension. It becomes loose. Okay, so how does it become loose? And this is the mechanism. Okay, let's have a look again. So, okay, I think it's better that we draw. Yeah, if I were to draw cell membrane, this is a proton pump. Okay, okay, This is cytoplasm. This is cell surface membrane. Okay, now this part here is the cellulose cell wall. Okay, cellulose cell wall. So just gonna make it thicker like this. Okay, this is the cellulose cell wall. You're talking about plants. Okay, so what happens is that the arrival of action potential. Okay, the arrival of action potential at the midrib or hinge, so will cause the pumping of this proton from here to here, cell wall. So this is low concentration to high concentration. Okay. So this will decrease the pH. Yeah. So remember, decrease in pH means it's very, very acidic. Okay, so it will break the cross link. Yeah, remember in cell wall there are cross link in cell wall between the cellulose microfibril, and that is weakened. Yeah, that is weakened, and so when it is weakened, the tension become loose. Okay, the tension become loose, and then when the influx, remember the influx of calcium, the influx of calcium okay, high concentration of calcium, low water potential. Okay, high calcium, low water potential, and so water will also enter. So this H2O will also enter. Water enters. Okay, so when water enters, this becomes turgid. Okay, so when the cells here becomes turgid, and that's when the convex shape is changed to concave. Okay, so let's have a look again here. So pH drops, cell wall crosslink broken. We have a look at the crosslink later on. Cell wall loosen, becomes flexible. Calcium pectate also dissolve. This is like glue between the a chemical nutrient that glues plant cells together, especially between cellulose cell wall. Remember, between cellulose cell wall there's a middle lamella, and the middle lamella has calcium pectate. Yeah, it also helps to keep the cell wall sturdy and rigid. So the acid, okay, does two things. The acid, the low pH does two things. The first one, crosslink broken, and then calcium pectate dissolves. And then of course the influx of the calcium, remember we know that already from the earlier one. Okay, the influx of calcium, because of the deflection of the hair, the first hair and the second hair, that will cause water potential drop, so water will enter by osmosis. Okay, remember this one here, the one I just draw, becomes turgid, and that's when it changes from convex to concave. Okay, so this is, I think, for your syllabus, I think that is enough, and that is the reason why, and that is the explanation why it is closed. The Venus flytrap is closed. Explain how the closure of the trap is achieved. So the closure of the trap is achieved. Of course, you have to talk about from the very, very beginning, which means the deflection of the first hair, and then the deflection of the second hair, which reaches threshold, causes action potential to move, spread over the lobe, and then reach the hinge. Okay, and then that when action potential arrives at the hinge or midrib, the pumping of protons occurs from cytoplasm to cell wall. This is important, cell wall. And then pH drops, weakens the crosslink in cell wall, cellulose cell wall, and dissolve calcium pectate. And because of the influx of the calcium, will lower the water potential, here, okay? Okay, so water will enter here, becomes turgid, okay, becomes turgid, and that's when it changed from convex to concave. And the video will show that it will shut at around 0.3 second. So it's quite fast, yeah. Okay. So this is the first stage, yeah. The first stage. The first stage means is the 0.3 second is the first stage. The second stage means complete closure, yeah, meaning totally sealed, and that's when the insects is when the insects is already trapped inside. For example, let's use a different picture here. Yeah, like this one here. This is already like almost closed, yeah, almost closed. But then this insect, when it's trapped, is like partially trapped, yeah, it will deflect more hairs, yeah, it will deflect more of the hairs, and that will cause more calcium to enter, and that will cause more depolarization and more action potential, and that's when it's becoming, then that's when this cilia will interlock, yeah, this cilia will interlock, or the stiff hair will interlock, and it will close the trap, yeah. We keep it, we close it completely, okay. And that is the what it's talking about here, the second stage. So the struggling, yeah, also it begins when it begins to struggle in such a limited space, it will deflect more of the hairs, and that can cause more calcium, yeah, more calcium to enter into cells, and more action potential occurs, and so that one, and that's when it's fully shut, okay. And what happens when it's already fully shut? So the influx of the calcium, this part here is important, yeah. I've just talked about step number 5 here, deflection of further sensory hair, further deflection of sensory hair due to restless movement of insect, and that will stimulate more calcium, okay. And that calcium will stimulate exocytosis of vesicle, and this is to release the digestive enzyme. So here is the this area here is the digestive enzymes, okay. So the influx of the calcium, because of the closure of the trap, okay, and then it will deflect more of this hair, which causes even more calcium, even more calcium, okay, to enter, okay. So that will stimulate vesicles, yeah, vesicles here, vesicles, to release the enzyme, digestive enzymes, by exocytosis. So the vesicle, which is inside the cell, will fuse with the cell membrane, and then it will secrete the digestive enzyme. It will secrete the vesicle will fuse, and then it will secrete the digestive enzyme. And that's how, and that's what it's stated here. Okay. So the syllabus is only up to here. You don't need to talk about number 7. Number 7 is the opening. Yeah, the opening, no need. It says only the closure. Okay, only the closure. So there's no need to think about the opening. But the opening is actually caused by more growth, which builds up the tension. Yeah, it says here, once the insect is digested, the cell on the upper surface of the meat grows slowly, and which will increase the tension, and then that's when the trap will open again. And this is not, you don't have to worry, you don't have to worry about step number 7, because it's not even stated in the learning objective. All right, so this is the answer scheme to a question about the Venus flytrap. Okay, how the Venus flytrap, how the trap is closed. Yeah. So again, mechanical energy. This is the deflection of sensory hair. Okay. Remember, it requires two sensory hairs to be deflected. Okay. The first hair will not reach threshold, but then deflection of the second hair will reach threshold and causes depolarization and then action potential across or the lobe and then down to the mid-rib. Okay. Reference to sensory hair, receptor, detect touch, cell membrane depolarizes. You see, you must have at least two hairs within, it says here, within 35 seconds. Most of the references between 20 to 30 seconds, or 35 seconds is also not that far away from 30 seconds. So action potential occurs. Okay, so remember, it's always depolarization and followed by action potential. Okay, when we talk about this is important. This concept is also very, very important in plants. Not in plants, synapse. Okay, let's talk about synapse. Synaptic transmission. Okay. The concept, yeah, that we are trying to so if we talk about the synapse last time, the arrival of action potential in the presynaptic membrane causes calcium to enter. Okay, and then the vesicle will fuse with this presynaptic membrane, release the acetylcholine. Acetylcholine binds to the receptor. Sodium channels open. Sodium enter. Then we say the entry of sodium will depolarize. Depolarize. What is depolarize? Depolarize means inside positive, outside negative. Okay, and then when it's depolarized, then action potential will occur. Yeah, so action potential occurs from one segment to the next segment to the next segment, yeah, from one node to the next node. So normally we don't say action potential occurs, we say it depolarizes the membrane and then action potential occurs. So it's step by step. Okay, so here you see membrane, cell membrane depolarizes. If the two hairs are touched, then action potential will occur. Okay, so normally action potential and depolarization, they actually mean the same thing, but in the case of maybe in plants, we have to be very, very careful, yeah, because the polarization may not necessarily cause action potential if it doesn't reach threshold. Okay, so it must reach threshold. The threshold is the second hair. This is the threshold. So the deflection of the second hair will reach threshold, and that is when action potential will occur. So it spreads over the leaf and then to the hinge. Then the pumping of hydrogen from the cytoplasm to the cell wall. Cell wall will loosen, cross-link broken, calcium pectate dissolved. Remember, the role of the lowering of the pH has two functions. Cross-link broken between cell wall, calcium pectate dissolved in the middle lamella. Okay, revise your lower 6. Okay, calcium enter. This is actually from the beginning, yeah, at the base of the hair. Remember this part here. Okay, this is the one that we talk about. The deflection of this hair causes calcium to enter into cells, and this is the one that we are talking about. Yeah, calcium enter, influx of calcium. Okay, where is it? It's this one. Okay, so that will lower the water potential in the cell, in the cytoplasm. So water will enter by osmosis. So it will expand, become turgid, and that's when it changes convex to concave. Okay, so normally in the exam, it may be only about 4 or 5 marks. As you can see here, these are about 15 to 16 marking points. Okay, marking points. There's a lot to actually memorize. All right, what do we mean by cellulose cell wall? The cross-link is broken. Cross-link between cross-link in cellulose cell wall is broken, and that's the meaning of the cross wall is the this one here. Cross-linking polysaccharide normally involves this one here, pectin and hemicellulose. Okay, so between the cellulose microfiber, remember this is microfibril, they are cross-linked, and normally they are cross-linked between this microfibril. So, for example, the pectin and perhaps the the green color here, hemicellulose, yeah. Okay, pectin and hemicellulose, and the acid, the low pH will weaken this cross-link and will make it loose so that it can expand, so that the cell can expand. Okay, that's the importance of it. Okay, the same thing here, but you don't have to talk about the expansion. This is more on the role of auxin. Okay, no need to look at the expansion here. As you can see here, here is the role of auxin. We will also be talking about this in the next learning objective. Okay, so don't worry about the auxin. But the same thing here, the mechanism is the same. Somehow the mechanism of auxin is also the same in cell elongation, as well as in the closure of the venous fly trap. You can see here the pumping of the protons, which lowers the pH, and it will break this cross-link. Okay, the cross-link will be broken, and so it will be easy when water enters, becomes turgid, it expands. It expands. When it expands, that's when the convex is changed to concave. Okay, now this is a good one here. This is, look at the strength of the stimulus. Strength of the stimulus. Let's have a look here. The red line, says it, the red line is the first stimulation of a trigger hair. See the red line here, when the first hair is deflected, it doesn't reach the threshold. It doesn't reach the threshold value here. Okay? But then if a second hair is deflected, the green line here, so if you read the green line is the second signal shortly after the first one. It says this is the green line. So if you look at the green line here, okay, the green line here, so if you, this is less than 30 seconds, okay, and that's when you see it reaches the threshold, exceed the threshold, yeah, and this is when the action potential will occur, okay, and will spread over the lobe, and then it will reach the hinge. And then if we look again, if past 35 seconds here, it is already past 35 seconds, this is the blue line. Okay, if you read the blue line here, the blue line represents a second trigger after the red one, but it is after the 35 seconds. So if after 35 seconds, this goes back, the time is reset. Time is reset. Okay. If what I mean by time is reset, meaning the time is equal 0 second. It goes back to square one for this one here because it's past 30 second. So this one becomes the first stimulus, the first hair deflected, because it's after 30 seconds. So time is reset to 0. So it will wait for another deflection. If that another deflection occurs within 30 seconds, then action potential will occur. So that's the meaning of that. So it's important to note that this is the first stimulus, this is the second, and all occurs less than 30 seconds. Okay, less than 30 seconds. If there's another stimulus, like another one, like this one here, after 30 seconds, then this becomes the first. It goes back to the first stimulus because it's reset. Yeah, it's reset. The time is reset because it's beyond 30 seconds. 35 seconds is too long already. Yeah. See here, the blue line is right here. The blue line represents a second trigger after the red one, which is just a split second too late. It's too late to trigger a response. Okay, to trigger a response. So if I can just repeat again, there are two scenarios here. This is definitely the first one. Okay, first deflection of hair. If the second deflection occurs less than 30 seconds or between 20 to 30 seconds, then action potential will occur here. Okay. If the second deflection, not the green one but the blue one, if this is the second deflection, second deflection, this is already too late. This is already too late. Because it's past the 30 seconds. So that means time is reset. That means this becomes the first one. The first one. Time is reset. Time equals 0 second. Reset. This one becomes the first one. So it will wait until the next deflection. Okay. Right. All right. What are the adaptations? This is covered already in the video. The adaptations of the Venus flytrap is that why it requires second hair to be deflected. Because this is already covered in the video. If you watch the video, because this hair is susceptible. It can be deflected by maybe dust. Or by maybe rainfall, rain droplets, which is less important, So if the first hair is deflected, it will not close because this can happen especially due to dust, due to rain droplets, and that's why it requires for the second hair to be deflected. Only then can it close, and not just that, even small insects, tiny insects, insects which are less in terms of nutrient, so the plants may not be interested to digest or to trap those very, very tiny insects, that's why it requires for a second hair to be deflected, so that the plants is not going to waste energy. Then the plants will only close when it thinks that the size of the insect is is good enough, yeah, for them to digest. So these are the benefits. The first one is already mentioned here. Okay, and then the gaps between the stiff hairs that form the bars allow very small insects to crawl out. Okay, so for example, this one here, these are the gaps. Okay, these are the gaps. So even though, let's say, if it manages to close, let's say the insect manages to deflect the first hair and then deflect the second hair and then it closes, if it's very very small, then it will be able to escape. Yeah, it will be able to escape through these gaps. Okay, and that will of course that will also benefit the plant, because it's not going to be worthwhile, yeah, for the plants to digest small insects. Okay, so when the insect escapes, then it will reopen back, the trap will reopen again. Okay, so these are the two adaptations, a very, very good adaptation. Adaptation C is not stated in the syllabus. Okay, so don't worry about the slides. The important thing is that you understand the concept. Yeah, the concept must be understood. When you come back, when you return, the notes will be given to you, because even since last week I have been printing the notes already. Yeah, so hopefully, so don't worry about the notes. I'll send you the soft copy of this, but don't print it. Yeah. Because you are entitled for the soft copy. Right, I think that's all for today. Yeah, so tomorrow, Wednesday, we'll be looking at auxin, yeah, the role of auxin, as well as gibberellins. Okay, this will be on Wednesday or Thursday. We'll see how it goes. Okay, that's all for today.